In this video, we are going to talk about polymerase chain reaction, or PCR. So I'm going to ask you, pause the video and think about what do you think about PCR, or have you heard about it before? Just pause and think about it a little. Okay, you got it right. Polymerase chain reaction is for amplification of the DNA, but not in nature human body, but also in the laboratory under the lab conditions and using the chemicals. What chemicals are necessary for polymerase chain reactions, or the devices that we need include thermal cycle. Thermal cycler is a device that is used for PCR chain reaction. The next is primers. If you remember what we talked about DNA replication in the past, you knew that primers are the fragments which are necessary for DNA replication. There are small fragments that will attach to the target of the DNA, which is going to become replicated, and will be recognized by the DNA polymerase. So the primer is attaching here, then coming after that is DNA polymerase, and the DNA polymerase start to add nucleotide continuously in a direction of the 5' to 3' in the DNA replication, if you remember. So these are the necessary elements for each PCR. Thermal cycler, I'm going to explain about that later. Primers, because they are necessary to attach to the target of the DNA target. And then free nucleotides, which is going to be added by the polymerase. So these are the necessary elements, and we are going to talk about this watching this video. So right now I'm going to click on this link and then watch the video together. Okay, here is the link after clicking, and you will see this page, and let's watch the video together.
A sample of chromosomal DNA, also called genomic DNA, can be used as the starting material for the polymerase chain reaction, or PCR. With PCR, an investigator can amplify a single copy of a DNA segment into billions of identical copies. The DNA segment of interest, or target DNA, is indicated in red. In addition to the target DNA, a PCR reaction contains several other ingredients. These include free nucleotides, DNA primers, and the enzyme Taq polymerase. The primers are typically about 20 nucleotides long and are complementary in sequence to the ends of the target DNA. Taq polymerase is derived from hot springs bacteria and can tolerate the intense heat of a PCR reaction. A PCR reaction lasts several hours and typically consists of 20 to 35 repeating cycles. A cycle begins by heating the reaction mixture to 95 degrees Celsius. The heat denatures the DNA, breaking the hydrogen bonds that hold the strands together.
Okay, this is the first step of PCR reaction, or the device as a thermal cycler is doing the first by denaturation of the DNA. So the two strands of the DNA is becoming separated by 95 degrees centigrade, and will be available for the next step, which is necessary for amplification or the replication of each strand. So let's continue.
After denaturing the DNA, the temperature is reduced to around 60 degrees so that the primers can form hydrogen bonds, or anneal, with their complementary sequences in the target DNA.
So if you noticed about this temperature, the thermal cycler is reducing the temperature into 60 degrees. Okay, so once you set up the thermal cycler for the PCR reaction, you will set 3 temperatures. The first temperature is for separation of the DNA strands by 95, and then after that it immediately reduces the temperature to 60 degrees, and at that temperature there will be the temperature which is appropriate for annealing primers. Primers are necessary for attachment to the target ends of the DNA, and the optimum temperature for annealing primers are 60 degrees.
Note that the primers and the target DNA follow base pairing rules. An adenine, A, pairs with a thymine, T, and a cytosine, C, pairs with a guanine, G. In the next phase, the temperature is raised to 72 degrees Celsius. Taq polymerase functions optimally at this temperature and begins polymerization.
Okay, if you notice, the third temperature is 72 degrees, and this is the temperature which is optimum for DNA Taq polymerase. This is a specific DNA polymerase which is called Taq, and it has the functionality over the high temperature degrees. Not that every enzyme can survive at this temperature, so that's one of the beauties and necessary things about this DNA polymerase, that can act and function at higher temperatures. And that's the reason that we have the PCR machine and the PCR technology, just because this DNA polymerase can act at these high temperature degrees.
Adding nucleotides to the 3' end of each primer attached to the DNA strand. After one complete cycle, there are 2 double-stranded copies of the target DNA.
Okay, pause the video and tell me, why do we have 2 copies of DNA at the end of the first cycle? Okay, you got it right. Because the DNA replication process or the amplification in a PCR machine has some advantages comparing to our replication in our body. And the advantage is we don't have the limitations of making Okazaki fragments at the PCR machine, because each DNA Taq polymerase can start from 5' to 3' direction and 5' to 3' direction, because the DNA already separated completely. You got the idea what I'm talking about? In our body, the DNA is not separated completely. So that's why as simultaneously the DNA strands are opening, and then the movement of the DNA polymerase would be only from left to right or right to left. So at the same time, it's not possible that doing 5' to 3' to make the directions for both strands. So that's why we have Okazaki fragments. But here, since the 2 strands are completely separated, it's completely free and easy and convenient for the DNA polymerase that can act from 2 opposite directions and then start to 5' to 3'. So here is 5' to 3' from right to left, and here is 5' to 3' from left to right, and that's why we have 2 copies of DNA at the end of the cycle 1.
The PCR reaction mixture contains many copies of the primers and an abundant supply of nucleotides to perform many additional cycles. After a second cycle, there are 4 copies of the target DNA. After cycle 3 is finished, there are 8 copies of the double-stranded target DNA sequence.
Okay, pause the video and think about it. Why do we have 8 copies after the cycle 3?
Note that only 2 of the double-stranded copies consist of just the target fragment. The others also include flanking DNA regions. As the number of cycles increases, the products consist of a greater proportion of fragments with just the target DNA. After 4 cycles, half of the fragments consist of just target DNA, and half of the fragments also contain flanking DNA.
Pause the video again and tell me, why do we have 16 copies of DNA after cycle 4? Find out the formula and let me know if you got it right, if you got the right answer.
With each additional cycle, the number of copies of our target sequence doubles. At the end of cycle 25, there are more than 33 million copies of this double-stranded target region.
Okay, can you calculate why do we have 33 millions of copies after cycle 25? Okay, if you thought correctly, you will get the same answer as me, because if we have one DNA at the end of the cycle 1, so cycle 1, how many copies do we have? So cycle 1, we have 2 copies. So this is cycle 1, and we have, in cycle 1, we have 2 copies. So this is cycle 1, and this is the number of the copies. In cycle 2, how many copies do we have? Each DNA create another copy, so that would be cycle 2. How many copies do we have? 1, 2, 3, 4. So in cycle 2, we have 4 copies. How about cycle 3? In cycle 3, again, each of them become double. 1, 2, 1, 2. 1, 2, 1, 2. Can you count? 1, 2, 3, 4, 5, 6, 7, 8. So at the end of cycle 3, we have 8 copies. Can you tell me what is the formula? Yes, you got it right. So the formula is 2 power n, in which n is the number of the cycles. So here is 2 power 3, so these are the number of the cycles. Over here, 2 power 2 and 2 power 1. N is the number of the cycles, and the number of the copies that we got at the end of each cycle. Okay, I'm going back to the PowerPoint file. We have already watched this one. So the next is PCR Virtual Lab. This is your homework. You're going to click on the link and then look at the virtual lab and do some experiment virtually by yourself. I am going to show you a little, and then you do the rest by yourself. Okay, this is the PCR virtual lab, and if you begin, so you will see some explanation over here. You are going to read, because we don't have time over here. I don't want to increase the time of the video. So you read by yourself and then go step by step next. And it's very nice. Shows that how, yes, a DNA is going to become. And this is how a DNA sample can be used for amplification using the PCR. And this is the PCR machine. This is what I already talked about. And what you are going to do is that, let's see how it works. So there is a micro pipette over here. You are going to take the drag the extracted DNA to the PCR tube. So you are going to take the DNA from here and then add to the PCR tube. So I need to use my okay, so this means that I didn't take it properly, so you need to use your mouse. Okay, so next, the next is this one. Each of them, so the first you picked up DNA, and then you need to add, and then add, and then you add primer 2, because primer, one from the left side of the DNA and one is for the right side of the DNA for the complementary strand. And then you need nucleotides. So you need nucleotides, and nucleotides over here. And then the next would be here. Now the PCR is ready, and go for the last step, which is taking PCR tube and put it inside the thermocycler. So yeah, so you can see that how each step inside the PCR tube inside the machine by denaturation and annealing, followed by extension, again denaturation at 95 degrees, primer annealing, and 72 degrees for extension of the nucleotides. And you see that at the end of cycle 2 you have 4 copies. And step by step from cycle 3 and again cycle 4 and cycle 5. Yeah, and then it's at the end of the PCR after 30 cycle. That's it. Thank you for watching.